The human body operates as a thermodynamic engine. It consumes fuel, generates heat, and constantly drifts toward energetic imbalance. Left unchecked, the internal environment would rapidly destabilize under the weight of its own constant metabolic perturbation. To prevent this collapse, the body relies on the endocrine system. This is a highly distributed network of specific organs, known as ductless glands, positioned throughout the anatomy. Rather than routing fluids through physical tubes, these glands secrete chemical signals, hormones, directly into the bloodstream. The circulatory system then acts as an open highway, carrying these chemical messages to distant target cells waiting to receive them. This messaging network exists to enforce homeostasis. Homeostasis is the active, high-energy maintenance of a dynamic internal equilibrium. Every second, the system must aggressively adjust to external and internal pressures to keep variables within a narrow, survival-critical range. When you walk into the cold, eat a meal, or stand up too quickly, variables like your core thermoregulation, blood glucose, and blood pressure threaten to spike or crash. Understanding how the body survives these shifts requires mapping two distinct biological mechanics: the localized, microscopic chemical structures of the hormones themselves, and the macroscopic negative feedback loops they execute. Systemic survival depends entirely on this unbroken chain of chemical communication. If the signals fail to deploy or fail to land, the biological equilibrium collapses. To understand how a hormone actually issues a command to a cell, we have to look at the primary physical barrier between the systemic bloodstream and a cell's internal machinery, the lipid bilayer of the cellular membrane. The endocrine system secretes two main types of hormones, the most common being peptide hormones. Chemically, these are constructed from proteins and amino acids, making them water-soluble. This animated diagram shows a cross-section of a lipid bilayer cell membrane. Because peptides are water-soluble, they face a strict mechanical constraint. They cannot penetrate the fat-based lipid membrane of the cell. The chemical structure literally bounces off the barrier. To bypass this physical limitation, peptide hormones dock into specialized surface receptors protruding from the target cell. The receptor's three-dimensional geometry is specifically shaped to recognize and bind with that exact hormone sequence. Once the connection is made on the surface, it triggers a second messenger cascade inside. The external hormone never enters the cell. It simply flips a switch on the outside that initiates a sequence of chemical reactions beneath the membrane. The counterexample to this is the steroid hormone. Unlike peptides, steroids are derived from cholesterol, making them structurally lipid-soluble. Because like dissolves like, this lipid solubility allows steroid hormones to diffuse seamlessly straight through the fat-based cellular membrane. They bypass the surface entirely. Steroids instead seek out intracellular receptors. They travel deep into the target cell, often migrating straight into the nucleus itself before locking into their designated receptor. This direct nuclear binding allows the steroid to physically interact with the cell's DNA, directly activating specific genes to manufacture new proteins. The macroscopic action a target cell takes is entirely dictated by the microscopic chemical makeup of the hormone arriving at its border. Zooming out from the individual cell, we find the origin point for most of these chemical signals located in the brain, at a central processing node called the hypothalamus. The hypothalamus acts as a strict neurological bridge. It ingests sensory data from the nervous system, like drops in temperature or spikes in stress, and translates that neurological data into actionable chemical commands. Positioned directly beneath the hypothalamus is the pituitary gland. If the hypothalamus is the strategic command center, the pituitary is the primary execution node, deploying signals across the entire anatomy. The pituitary is divided by function. The posterior lobe acts as a storage and release facility for hormones produced by the hypothalamus, like antidiuretic hormone, which regulates water balance. The anterior lobe, however, operates as an active manufacturing center. It produces its own systemic signals, such as growth hormone, which dictates cellular division and tissue repair. We can trace this command logic down the hierarchy. The anterior pituitary secretes a hormone into the blood that travels down the neck to activate a specific peripheral target, the thyroid gland. Once stimulated, the thyroid executes its localized responsibility. It synthesizes hormones, which require a steady supply of dietary iodine to regulate the body's basal metabolic rate, dictating exactly how fast surrounding tissues consume energy. This physical, top-down hierarchy allows the brain to constantly calculate energy demands based on environmental stimuli, and then subconsciously dictate energy expenditure across millions of independent cells simultaneously. Peripheral organs also operate their own localized networks. The pancreas is uniquely structured for this. It releases digestive juices into physical ducts, but it also contains distinct endocrine tissues that release hormones into the blood. The pancreas functions as a perfect biological control loop, utilizing negative feedback. In a negative feedback loop, a system detects a deviation, deploys a countermeasure, restores the baseline, and then shuts the countermeasure down. This chart maps a negative feedback loop, plotting blood glucose concentration over time. The central, flat line represents the required homeostatic baseline. After a meal, the charting line begins rising sharply above that baseline. The pancreas detects this upward deviation and deploys an immediate chemical countermeasure, secreting the peptide hormone insulin into the bloodstream. Insulin binds to target cells, forcing them to absorb the excess sugar out of the blood. This mechanical extraction drags the systemic level back down. Once the baseline is restored, insulin secretion shuts off. If the body goes too long without food, the variable drops below the baseline. The pancreas detects this downward deviation and deploys an opposing countermeasure, a hormone called glucagon. Glucagon targets the liver and muscle tissues, forcing them to eject their stored glucose back into the circulatory system, raising the blood sugar until the required baseline is restored. Homeostatic stability is never static. It is an aggressive, constant correction of continuous deviations, managed through the antagonistic interplay of precise chemical tools. When we map out these control loops, endocrine diseases stop looking like arbitrary ailments. They reveal themselves as distinct mechanical failures at highly specific nodes within the signaling network. Take diabetes mellitus. Clinically, it is the inability to maintain normal blood sugar levels. Mechanically, it is a structural collapse of the glucose feedback loop we just mapped. This split-screen flowchart visualizes two distinct points of failure. Type 1 diabetes, shown on the left, is a primary output failure. The pancreas itself physically loses the capacity to manufacture insulin. The signal is never sent. Type 2 diabetes, shown on the right, is an interface failure. The pancreas continues to output insulin perfectly well, but the specific surface receptors on the target cells become resistant, physically rejecting the connection. This resistance blinds the cell to the chemical command. Even though the bloodstream is saturated with the hormone, the biological hardware cannot process the message, and the feedback loop remains paralyzed. This logic applies backward to the central command hub as well. Consider a structural failure in the anterior pituitary. Acromegaly is triggered by a breakdown in the pituitary's negative feedback mechanism, resulting in an unchecked, continuous overproduction of growth hormone deep into adulthood. Because the signal instructing tissues to multiply never shuts down, the macroscopic consequence is severe. The continuous hormone exposure forces the expansion and enlargement of bone structure, clearly visible in the hands, feet, and facial bones. Moving back down to the peripheral thyroid gland, we see what happens when a gland experiences a chemical supply chain failure. The thyroid requires iodine to synthesize its metabolic hormones. If a person's diet lacks iodine, hormone production halts. The thyroid detects this deficiency and attempts to compensate by working harder, eventually causing the entire organ to physically enlarge, a condition known as a goiter. Large-scale, observable systemic pathologies are not random. They are direct readouts of localized, microscopic breakdowns in the underlying biological hardware. The endocrine system operates at a massive scale, bridging the gap between molecular kinematics and macroscopic organ control loops. Its function is bound by two strict realities. Physiological action is constrained by chemical structure, whether a molecule is a lipid or a peptide, and survival is dictated by the structural integrity of the network itself. As we've seen, homeostasis is a relentless, continuous calculation. It requires constant output, constant monitoring, and constant countermeasures. Every step you take, every calorie you burn, and every drop in temperature you survive relies on this invisible back-end regulation. Profound health and devastating disease hinge entirely on a chemical message successfully moving from gland into the blood and securely into a receptor. The human body functions as a highly coordinated control system, surviving only by mastering its own internal chemical instability.